Welcome to the United Leukodystrophy Foundation's first live webinar. We hope that you enjoy tonight's content. Thank you so much for joining us. And I would like to remind everybody that we are recording this session. So if you wish not to have your voice or your face on screen, please keep your microphone muted and your camera off, as we will be posting the recording to YouTube. So please keep that in mind if you have privacy concerns. This is being recorded. We will post it to our ULF office YouTube page tomorrow morning. And we are having 2 more live webinars. The next one coming up in November is based on siblings of leukodystrophy. We have 2 siblings with a brother that has the type MLC, and they will be presenting on what it is like to be a sibling of leukodystrophy, and just talking about their experience and providing input. They'll be answering questions. So we encourage everyone to attend. If you're a parent, if you're a friend, if you are a sibling yourself, the presentation is open for everyone. It's meant to be a very honest and open discussion about the sibling experience being affected by leukodystrophy. And then in December, we will be hosting a self-care through the holidays webinar, which is being led by a social worker that will help the families hopefully learn how to cope with stress during the holidays, learning how to enjoy the holidays while you have all of this craziness going on in your life. If you're going through newly diagnosed, if you're just having a hard time this year, we're hopeful that that session will allow our families to hopefully navigate the holiday season a little bit more easily. And then we will be announcing our 2022 schedule by the end of the year. So please keep an eye on our website and our Facebook page to see what craziness we have in store for everybody next year. We will be starting up the support groups once again. But we will be alternating between support groups and live webinars next year, each month. And then of course, the month of June is when we will have our conference, which we do fully intend on having that be in person next year. And we do hope to have a virtual aspect. We will try to stream some of the sessions. We're not sure if we're going to be able to get all of the sessions streamed or recorded, but we are going to do our best. So without further ado, I would like to introduce Rachel Logan. She is a genetic counselor with Children's Healthcare of Atlanta. And please keep your questions handy if you want to jot them down, if you want to write them in the chat. Rachel will be answering questions at the end of the presentation. Otherwise, we do ask if you please keep your microphones muted. You can feel free to turn your camera on if you wish, but do please keep your microphone muted. Thank you so much for joining us, and ready when you are, Rachel.
Wonderful. Thank you so much for inviting me. I've really enjoyed working with the United Leukodystrophy Foundation in the past, and it's really wonderful to be able to have organizations like yourselves that are doing this work in the community, and I appreciate you having me today. So today I want to talk about genetic testing for the leukodystrophies, just because that process and question comes up a lot. It's something that I do a lot as a genetic counselor. And then I do want to talk about just an introduction to the science behind gene therapy. So I'm not going to talk about any specific gene therapies or any specific trials, but I do want to kind of define some terms. So when people talk about gene therapy, or you see something online about gene therapy, I want to kind of empower you with kind of what some of those terms mean. So just move this here, got Keely's little face. Okay. So this is, this lecture is not medical advice. If you have leukodystrophy or you love someone who does, this isn't treatment advice or diagnostic advice, but just kind of some general themes. I don't own any of the pictures I use. Some of them are made, but most of them I don't own any of them. And then I work at Children's Healthcare of Atlanta and don't have any other financial conflicts of interest to disclose. So, of course, we have to start a lecture on genetic testing with a picture of DNA. I had a professor in college that believed that you should show the pictures of the molecules if you can, but I don't want. This is probably the last time I'll show actual molecules here. But so in all of our cells, is most of our cells, just anyways, are our DNA. So our DNA is our body's instruction manual. It's our instruction manual for forming during pregnancy and functioning throughout life. It's packaged into structures called chromosomes. We get our DNA from our parents. And like I said, our DNA is our body's instructions. And so, and when we get down to that, ultimately the job of our DNA is to be the instructions for forming proteins. And I know sometimes people think about proteins like, the meat in the sandwich they had for lunch, but proteins really are the backbone of our body. Our body is made up of lots of different types of proteins and fats and sugars and water. And the job of our DNA is to create proteins. And so the way that it does that is through sort of an intermediary substance called RNA. So our DNA is essentially copied by the cell into something called RNA. And then that RNA is used to be the kind of the guideline for making lots of different types of proteins. So there's some proteins that are made only by some cells, and there's some proteins that are made only at certain times. And the complexity of sort of when and where and how much is just a very sophisticated dance that our body does without us even thinking about it. To simplify this, I think an analogy is sometimes useful in understanding what all these components are, and I'll kind of refer back to this analogy quite a bit. But our cell is kind of like a kitchen. Its job is to make lots of different foods and proteins to keep us going and to keep us healthy. Our DNA is kind of like that central recipe book. So if you've got a big, heavy recipe book from a grandparent or something like that, that's kind of what our DNA is. It's passed on from generations. We get it from our family members. And it's really kind of that central point of the kitchen, knowing kind of what to make and when. But it's big and it's heavy, and we may not want to lug it around when we're in the kitchen and we're cooking. And so our RNA is kind of like taking a recipe or kind of writing something down and telling you what you're going to make. Not something that you're maybe going to keep or something you might put back into the DNA, but it's more of kind of like a short-term, smaller reference for making that day. And then our proteins are all of the different foods that we can make in our kitchen. So genetic testing. Genetic testing for leukodystrophies is a central part of the diagnostic process. So leukodystrophies are genetic conditions where the body's instructions for the brain and nerve tissue aren't working properly. So at the end of the day, it's caused by a difference in the spelling of that person's DNA that causes the body not to be able to make that recipe. For most of these conditions, the testing is done through genetic testing, but there are also some disorders that we can do some diagnostic testing with enzymes or biomarkers, and that's kind of looking more on kind of what did the recipe make side of things, and I'll kind of compare and contrast when we might use those. I think it's important to define the context for genetic testing. So genetic testing can happen for a lot of different reasons. There's the main one that I think people think about when they think about genetic testing is diagnostic testing. Diagnostic testing is saying, okay, does this person have whatever disorder it is? A subset of that would be predictive testing, which is where there's a known diagnosis in the family and somebody's undergoing testing to find out whether they may have that condition or not. Another form of genetic testing is something called carrier testing. And carrier testing is when somebody is not looking to find out whether they have the condition, but rather whether they're at risk to have a child that also has it, or whether they may be at risk for health complications related to their carrier status. Something that I put on here that I think oftentimes people don't realize, and it happens more than other people think, is something, there isn't really a great name for it out there, but I've named it consultative testing. And that's when we do genetic testing on the relatives of somebody to consult on behalf of that person. So occasionally we find a difference or a variant of uncertain significance in the DNA, and it's useful to test other family members to see, do other family members have this? Do they also have the disease? And so occasionally we'll recommend genetic testing, and it's not really for the person that's doing testing, it's for their family member that we're trying to figure out what's going on with them. So one of the questions that I get a lot is, kind of why undergo genetic testing? What is the purpose of it? So for diagnostic testing, it's helpful to provide a precise diagnosis. It's useful to kind of know what it is, and it helps us on sort of the medical side of things be able to answer your questions as best we can. It can help determine eligibility for therapy. So oftentimes for certain medicines or therapies, you have to have a confirmed diagnosis of leukodystrophy in order to qualify for it. The same goes for clinical trials. So for a lot of clinical trials, somebody oftentimes has to have a confirmed genetic diagnosis to participate. It helps us feel like, we can answer your questions and give you an idea of what the disease is going to look like. It can provide an opportunity to connect with other people with the same condition. I know I'm a little bit preaching to the choir here, but leukodystrophies can be quite a big umbrella, and there's a lot of different types. And so somebody's experience with a specific leukodystrophy might be different from another person with another leukodystrophy. And so knowing exactly which one it is can be helpful in sort of connecting with folks with kind of more precisely what they have. And then it also can estimate risk for other family members. So if there are other folks in the family who may think that they may be at risk for the condition themselves, or who are perhaps interested in having children and would like to know the chance for another child to have it, it can be useful to provide more specific risk estimates to different family members. And just to so predictive testing, we kind of talked about this before, but it's when there is a specific known genetic diagnosis in the family and the person undergoing testing is at risk for the condition, and the person is younger than the age at which first symptoms arrive, or they do not yet have symptoms. This is something that as a genetic counselor, this is our kind of space that we often help families with. So this might be a reason somebody would meet with a genetic counselor to talk about kind of who should, when is predictive testing a good idea? What are all the things that somebody might not think about when they're doing that process? And so there can be some benefits to it, but there can also be some drawbacks to it. And, is now the right time is an interesting question with that. Another reason why somebody might undergo genetic testing, like we mentioned, is carrier testing. So it can be useful to learn about the chance for somebody to have a child with a leukodystrophy. There are some reproductive technologies out there that require that you have sort of an identified genetic variant in the family. For some conditions, and I would say most X-linked conditions fall in this category, but there can be some health risks related to carrier status. And I think we oftentimes haven't done a great job of recognizing a lot of the symptoms that the female relatives of male individuals with X-linked leukodystrophies experience. Occasionally, carrier testing can be recommended to determine whether a person is eligible to be a donor for their family members. When we talk about things like stem cell transplants, there are some conditions where if you're a carrier for that disorder, it may make you eligible or ineligible to be a donor for stem cell transplant. A big caveat with this is oftentimes I get a lot of questions from parents about carrier testing in the unaffected siblings of their child that has a leukodystrophy. At our center and sort of nationally, there are lots of big guidelines about carrier testing in children. It's really not the right time to do that. If it's being done because they're at risk for the disease, and there's things that we may be able to do to help them or to determine whether there could be a donor for transplant, those are kind of other reasons why we might do it. But if it's really just to know whether they're a carrier, it's typically not recommended. Kids at that point should be worrying about who they sit with on the playground and not, what are the reproductive, questions about having kids and all that. So it's often really not the right time. So when it comes to kind of testing strategy, I want to acknowledge that the genetic testing process is often long, it's complicated, it can be expensive, it often is sort of a tiered approach. So, you know, if somebody is kind of in the middle of that process or they've been through it before and it feels like it kind of takes multiple steps, it's not necessarily because your healthcare provider doesn't know what's going on. It's just because sometimes it's just a multi-step process. And there's different strategies about when we might use one test versus another. So that's something I work with neurologists. That's something that myself and the neurologist, when we're looking at a child who we think might have a leukodystrophy, I work with children. But we often have a conversation about what's the right strategy for this particular patient to try to maximize, you know, the time to get to a diagnosis, the amount of clarity, you know, sometimes with their insurances. What do we know about their family members? What do we know about their symptoms? So we want to get the diagnosis in sort of the most cost and time effective way. And sometimes that involves one strategy versus the other. So let's kind of go through what all those are and some pros and cons. So single gene testing is, if you've gone through this, you probably knew hopefully exactly what was going on. So this is really when we're testing for one condition. So we'll do this when there's kind of a strong clinical suspicion for a particular condition, or if there's a family member that also has it. In these cases, the testing is a little bit more of a yes or no. So these are ones where somebody has some clear symptoms, we're pretty sure we know it is, and we're kind of doing more confirmatory testing. There's still a chance for some uncertainties with this type of test, but it's typically, oftentimes families kind of know exactly what they're being tested for and not. So a gene panel is where we take essentially about 100 or sometimes up to about 1,000 genetic conditions that all have one thing in common. So often it'll be something like leukodystrophy as a common feature. So there are some leukodystrophy panels out there. And the goal with these panels is that they lump a lot of conditions that can present similarly all in one test. And the turnaround time on them can be about maybe a month or two. And so those are sometimes a nice kind of medium approach when we're thinking about, we have a strong suspicion for a leukodystrophy, but not one in particular, just because there can be so much overlap between them. And then exome or genome sequencing, there are differences between those two. I'm not going to get into them too much unless somebody has questions. I'm happy to. But exome and genome sequencing are very broad genetic tests. So these are ones where we look at not all, but almost all of the DNA that somebody has that we know is known to be associated with any sort of medical condition. And it's one where we often use if there's sort of a complex history, which may suggest that it's leukodystrophy, but there are kind of some overlapping features that might suggest it could be something else. Oftentimes too with leukodystrophies, we're learning more and more about them. And so doing a broad approach like exome or genome sequencing allows us to kind of take advantage of the most recent research on those things. And so often genes that we don't know a ton about are more likely to be included on an exome and genome analysis versus one of those panels, where those are really more well-described conditions. Or perhaps somebody has had some testing in the past that's been normal or non-diagnostic, then we might think about doing something like exome or genome sequencing. The drawbacks to this one are that it typically takes a little bit longer, and due to the complexity of it, we often recommend including samples from the individual's parents because we get half our DNA from our mom, half our DNA from our dad, and so including some context to that individual's large amount of genetic data does help us sift through it a bit, but sometimes those folks aren't available or they're not wanting to participate in it, and so that's often something that comes up as well. And then there are some forms of leukodystrophy that can be tested for by looking at specific fats or proteins in the body. So these would be more like metabolic tests or biochemical tests or biomarkers. This can often be a blood or a urine sample. And I listed some examples here. The benefit to these is that they tend to be pretty specific. They tend to be a little bit faster than genetic testing. Occasionally, they can give us a little bit of information about the disease progression or kind of how severe it might be, sometimes not, but and oftentimes the genetic and the biochemical analyses are kind of complementary. So we want to see if we're suspicious somebody has metachromatic leukodystrophy, for example, we want to see that their genetic testing was abnormal, their enzyme levels were low, and then maybe they had a buildup of certain substances in their urine, and that all kind of goes along with our some supportive evidence that this is indeed the right diagnosis for that patient. So when it comes to genetic testing results, so there is no one person that has the normal DNA. We all have differences in our DNA. That's what makes us all different from each other. And so there is a good bit of interpretation that goes into analyzing somebody's genetic information and coming up with a result. So typically the results fall into one of three categories. So there's a positive result. Some words that get used with this are sometimes the word pathogenic variant or likely pathogenic variant. Occasionally some older terminology is something called a mutation, but we don't love that term. And that would be, okay, this is a positive result. This person has this type of leukodystrophy. It's a pretty clear, this is what's going on kind of result. A negative result is when the testing, so whatever test we ordered came back normal. So there wasn't anything identified on there. And a negative result, I often tell patients, is kind of means one of two things. So it either means that this person, truly does not have any of these conditions, then they don't have a leukodystrophy. Or a negative result could mean that for whatever reason, the test just wasn't able to pick up what is in fact going on with this patient. And it could be because that just wasn't good enough, or there was, something little that was missed in the analysis. And so just because of the complexity of these tests, they're not perfect. So there is no one like, oh, we're doing the genetic test and this is going to test for everything. That doesn't really exist. So oftentimes we have to do it in sort of a tiered approach, or sometimes we have to take kind of multiple looks at it to try to figure out what's going on. So, and then an uncertain result is the one that I often spend the most time with. And so an uncertain result, if you've heard of a VUS, or some people call it a VUS, and that's a variant of uncertain significance. And everybody has variants of uncertain significance in their DNA, everybody. And essentially what that means is that the lab found a difference in the DNA. So they say this is a little different than what we've normally seen. But it's not one that we have enough evidence to say for sure that this is a positive result, or for sure that this is a negative or normal result. So us as physicians and as genetic counselors like myself, our role on the team is just to take a look at these, and we'll give you a sense often for what we know and what we don't know about that. Sometimes it's just that it's really rare and we haven't seen enough times to be absolutely certain, but there's a lot of things that kind of line up with it, sort of feeling this is probably it. Or, there's just not enough here. We really don't think this is it. So on the clinical side of things, it's often us that kind of take it the final mile from the lab and try to correlate it with what we're seeing in that person's medical history. But sometimes uncertain findings, they're uncertain, and they're going to stay uncertain, but they don't stay that way forever. There's new research happening all the time, and eventually variants of uncertain significance or uncertain results, they get resolved over time. Just sometimes it takes a while. So another role I have on the team is just when we know a patient has had an uncertain genetic finding in the past, every time they come into clinic, I just kind of take a look at it again and see, is there any new evidence that's come out? Is there anything more that we've learned that might help us kind of push that needle one direction or the other? And this is just kind of a general, but the genetic testing process is hard. If you've been through it, or you've been through it in the past, or you're in the middle of it, I just want to acknowledge that it often takes a long time. There's a lot of acronyms. The results often aren't, it's not like normal blood work. It's not like getting, just something quick checked. It's often a lot more complex than that. It's often occurring at a time of a lot of uncertainty for people. Often it's kind of this roller coaster of like, well, we did the one test and it came back normal, and now we're doing another test. And it really asks for your vulnerability. I think our DNA is what makes us who we are, and we don't take for granted, we try not to take for granted that, we're asking to analyze something that is deeply personal to you and to your family members. And I think a lot of times the reason I get the, why should I do genetic testing question, and it often comes from a place of some resistance and distance. We're like, well, what's your really good reason to convince me to do that again? And I think where that comes from is especially for people who are older, who have been, they have a diagnosis of a leukodystrophy, but not a specific one, or they went through the diagnostic testing a long time ago. There's a lot of work that people have done. The initial diagnostic process is really a difficult time. It can be really devastating. And when somebody is not able to tell you exactly why, it can be really hard, and it can sort of break that trust with the healthcare provider. And it also, a lot of times it's just expensive, or and there's a lot of work that people go into sort of, what they know and what they don't know about their disease or their loved one's disease. And I think some of that resistance to jump into that again is just kind of shows you how far someone's come with their journey with their particular disorder or their family member's disorder. And I just, with humility, want to say that, we've been doing a lot of work in the genetic testing space for the last, many years, and it's gotten, and we've done a lot of work on it. It's gotten a lot better. It's a really exciting time to be doing this. And so I'd like to just, extend a hand and say, if you haven't gone through that process for a long time and it's still something that's an unknown, we'd like to show you what we've been working on and how, you might benefit from it. So with that, I want to kind of transition to a little bit of science behind gene therapy. This is a really exciting area of research. There is a lot of clinical trials. I just looked it up today. There's a lot of clinical trials out there that are involving gene therapy. It's been around since the 90s, with some ups and downs. And so it's a cool science, but it's also kind of weird. So I want to go into what it is exactly. So gene therapy is a process by which DNA is used to help the body function as if the person was not born with that genetic condition. So we essentially are adding to or modifying that person's DNA to change it to be more like a person who doesn't have the disorder. And so it's when DNA itself is the therapy. So if a genetic condition is caused by somebody's DNA that's not working properly, gene therapy is sort of answering that with DNA itself. There are multiple ways that this DNA can be delivered to the body. Viruses are the most common one. So I'm going to mostly talk about viral mediated gene therapies today. There have been some other ones that have been in the research space, but they're not as common. And so most of the work's been done using viruses to deliver this DNA. So you might be asking yourself, in the middle of a global pandemic, well, why would we be using a virus? And that would be a very good question. So I want to just do a little bit of background just to say that so virology is this like totally other field typically to what we do in genetics, but there's lots of different types of viruses. So this is just a schematic from a Nature article I saw that showed different types of viruses. The ones that are often used in gene therapy look like this one on my right here. So you're going to use this little cartoon to depict the viruses that might be used in gene therapy, but just to say that there's lots of types out there, and there's some reasons why we chose the ones we did. Let's see how I'm doing on time. Doing all right. Okay. So I think it's useful to do a little bit of a background on how do viruses work. So this is, if you remember this little blue blob here from our initial cell diagram. So this is representing a cell here. So, and we're moving from left to right. So, and we're close to Halloween, so I figured I could use a little vampire imagery here, and it wouldn't be too far off theme. So the way that viruses work generally, and they all have their own kind of way that they work, but generally what happens is a virus gets into our body, and it knocks on the door of our cells and says, hey, I'm the pizza delivery guy, and I would like to come in and bring you a pizza. And the cell says, okay, great, I love pizza, come on in. And so the virus then gets into our cell, and it interacts with our DNA and says, it kind of comes into our kitchen and says, whatever you were making before, stop making that. I now want you to make more of me. So it kind of hijacks our cell's machinery. It delivers its own DNA. And so it comes into the cell, it delivers its own DNA and tells the cell, you're going to work on making more of me. And then it essentially kind of takes all the cell's resources to make more of itself. And then eventually that's too much for the cell. The cell dies off and it releases a bunch of viruses that then go on to do the same thing. And then when you cough or you sneeze or, whatever, because you have a bunch of viruses that have replicated in your body, they may be passed to someone else. And so that's generally how viruses work. So they get into our cells, they hijack them, and then they use it to make more of themselves so they can spread. So gene therapy is a little bit like this, but not quite. Gene therapy is essentially saying, okay, let's look at this process. We like the idea that a virus might be able to get into our cells and interact with our DNA and potentially change it or add to it. But we don't love the idea of it replicating and getting out and being evil. And so what scientists have done is they say, okay, so we like the first 2 steps of the process. We don't love that second part. And so we do is we create viruses or we modify existing ones so that they behave, so they're kind of essentially using their superpowers for good. So it looks like this. So now we have our virus for good here, and it's wanting to come into the cell. It comes into the cell. It delivers its DNA, which are these little, I don't know if you can see my screen here. These little squiggly lines here are DNA. And it comes into the cell, it delivers that DNA, and then it breaks down. So it says, okay, that was it. That was my job, and my job is done. So it does not replicate. So it does its job, and then it's done. And then afterwards, you can see that cell has its DNA changed from orange to purple. And that's kind of the general idea behind gene therapies. I can't get it to switch. Lost my cursor. There we go. Alrighty. So there are a couple of different approaches that we use. So some terms that you'll see when folks are talking about gene therapy. So there's the integrating type, which is where the virus goes in and it goes to that sort of central recipe book, those recipes that we got from our family that are in every kitchen in our body. And it goes in there and it modifies it in some way. So it says, this is the DNA that is in all these cells, and I'm going to go in there and I'm going to change it. The non-integrating type is essentially where the virus goes in and it just delivers some extra DNA. So it puts essentially another recipe book on the shelf, one that's maybe a little smaller, and one that's perhaps able to make up for what's not in that main recipe book. It doesn't change the cell. It doesn't change. It essentially doesn't go into the sort of that central recipe book that we call the germline, but it just kind of adds a supplement to it. So there are some certainly pros and cons to both strategies. So integrating is nice because it gets into all the cells. It kind of goes straight to the source. Some downsides are that, that's kind of our, those are recipes that we've had for a long time. They're really well balanced and they've been, kind of perfectly in tune to be just right for our kitchen. And so, changing that is just a little riskier. It's also something where, but it then can be used by other cells. Whereas non-integrating, it doesn't kind of mess with that, it just adds to it. But then, if you move, what I mean by that meaning is that the cell divides, is that it's often not able to take those extra recipes with it. And so that works a little bit better for cells that aren't turning over time. So our cells kind of have this, some cells are turning over and dying and creating new ones all the time, so like our skin or our stomachs. Things like our nerves and our brains, not as much. And then there's ex vivo and in vivo. So ex vivo is when essentially a tissue is taken out of a person. It's treated with the virus. So we take these cells out of, so typically this would be used anytime we would, so this sort of approach could theoretically work for any condition where stem cell transplant has been effective in the past. So this is essentially we take the bone marrow out of a person, We use the virus to change the DNA that's in those cells, and then we put it back into that person. And then our bone marrow, the job of it is to create blood. And then over time, for the rest of that person's life, theoretically, their blood is essentially expressing the DNA that has a working copy of that gene or that recipe. And then in vivo is essentially when the vector or the virus is delivered directly into the person undergoing therapy. So that would be, and it's kind of systematic, so it goes into all their cells. So oftentimes that's an infusion. So it's one that they have the virus and they give it to the person through their blood, and then it's sort of a one-time thing, and it's delivered kind of to all of their cells. And I sort of kept these on right and left because oftentimes the integrating approach is used with ex vivo therapies, and the non-integrating approach is often used with in vivo therapies for kind of various reasons. And then some kind of virus terms that you'll see get thrown around. So lentiviruses and adeno-associated viruses are sort of used for different purposes. So lentiviruses, and again, kind of keeping with our columns here, lentiviruses are often used for ex vivo therapies. They're really good at kind of going in and interacting with that DNA that we were born with. It can go in and kind of edit or modify it in some way. Whereas adeno-associated viruses are pretty good at delivering that kind of extra copy or sort of supplementary recipe, but it's not as good for at going in and sort of modifying the DNA that we were born with. And so there's sort of pros and cons to both strategies. And so that's why lentiviruses are good for one approach, but not the other. Adeno-associated viruses are used for one and not the other. The reason why adeno-associated viruses and lentiviruses are used is because there are very few human diseases, especially for adeno-associated viruses, that are caused by AAVs. So the body often doesn't recognize them as a threat. They also are like pretty good at their job. So some viruses are kind of clunky and they're not that good at their job. Lentiviruses and adeno-associated viruses are generally pretty good at their job. And our job meaning kind of delivering that DNA or getting into cells. And so that's why we've chosen these. But there are some other ones that have been kind of tried out, and that's kind of a evolving field of research. Our little cartoon figure down here kind of looks like a lentivirus. That's where that came from. So in terms of strategy, so designing these therapies is something of a task of figuring out, okay, so what is the issue here? So what about this person's difference in their DNA is causing their disease? And so it's important that we understand, at the root of it, why that particular variant or difference in their DNA is causing that person's disease. So if we know that, then we can sort of target our therapeutic strategy appropriately. So sometimes there's a repair strategy. So we say, okay, this person, there's a misspelling here. It's causing the gene not to be able to do its job properly. So we can just go in and repair it. Sometimes it's because that gene is missing. So it's, maybe it would be working just fine, but it's not there. So we go in and supplement a copy that's not there. Sometimes the gene is in there and it's actually causing problems. So like the protein that it's creating is kind of interfering with other processes, or it's clumping together and causing problems that way. And so sometimes it just makes sense to knock it out. And so the thought maybe would be to just say like, okay, well, this gene's causing problems, so let's take it out. And then the other one is just kind of regulating. So so much of biology is a really well-balanced dance between all of the different millions of things that are happening all the time. And so sometimes it's that particular variant is causing the DNA to be used too much or too little, and sometimes we just need to kind of tweak how much of it is being used in the body. And so that's kind of what the regulating strategy is for. There are a lot of challenges with gene therapy. It's been a really exciting area of research for a long time, but it's getting it just right is really the thing that scientists have been working on the most. So just that really precise design of the therapy is what is so challenging. So I kind of get to this in some of the text, but just how much of that, so expression refers to, when the body goes in to make those recipes, we want to make sure it's made enough, but not too much or too little. We want to make sure it's made in the right places. So it doesn't really matter if you're expressing it somewhere, but it's not getting to where it needs to go. We obviously don't want the body's immune system to attack the virus or just to attack in general any sort of extra thing we put in there. This is less so nowadays, but we obviously don't want that virus to get out of that person and then start kind of going in places where we don't want it to. The timing of therapy is important too. So for some of these therapies, before somebody has symptoms or even earlier in their disease, that it may be more effective than later on. So it just depends on kind of the timing of that disease and what our opportunity is for intervention. So for some of the therapies, the target study population is very young individuals. We also don't want to, modifying our DNA as a sensitive thing. And so we want to make sure we do it with precision and intention, and we do it just right, because we obviously don't want our DNA to be modified outside of that. We want to do it just where we want to and not anywhere else. Here's some of my references. I thank you all for coming to join us tonight and to kind of talk about what's been going on and for genetic testing and then also gene therapy. I'm happy to take any questions. There were some really good questions that were submitted ahead of time. But thank you all again for having me.
Thank you so much, Rachel. That was an amazing presentation. We do have a couple of questions waiting in the chat. And so if anybody else wants to go ahead and put your questions in the chat, please feel free to do so. Or if you wish, you can unmute yourself and ask the question live as well, whatever you're most comfortable with. But first, let's start with what are the drivers of long turnaround times in regards to the different types of genetic testing? And why can't these tests be done in a day or a week, etc.?
Ooh, that's a really good question. So there's essentially what happens is when your tissue arrives at the lab. So oftentimes that's blood, but sometimes it can be cheek swabs or mouth swabs. What we have to do is we have to pull the DNA out of that sample. That takes some time. So we have to kind of pull it out. We also have to amplify it in most cases. And so what that means is that we have like tiny amount of DNA in our in sort of the blood or the whatever sample that was submitted. And so we have to go through a process of making sure we kind of copy it to make sure there's enough to look at. And then we have to go through and actually sort of read all the base pairs. And then there's an analytics part of it. So there is often a scientist at the lab that has to go through and kind of check off, manually go through all the differences in our DNA. So for something like exome sequencing, there can be something like 10,000 different differences in someone's DNA that pop up on there. And somebody kind of has to go through and filter through all those. So some of it is just because the actual setup time for the lab test takes a while. So there's steps that have to happen in a time sensitive way. And then sometimes it's just the analysis takes a while. And so so you wouldn't want somebody to kind of rush through that part. But yeah, so we've come down quite a bit. It used to be see ya in 6 months. This takes forever. For some labs, we've gotten it down pretty quick, but even for, there's another hat that I wear is neuromuscular. And so there is a gene therapy for one of the diseases we see called spinal muscular atrophy in a neuromuscular clinic, and getting that done is really time sensitive. And we've only been able to cut that down to maybe a week. So it takes, and we're all sitting around waiting on those. Nobody's ever waiting around for me. And I'm I'm sorry, it takes 5 days. It's the fastest we can go. And everyone has the same mutation for that disease for the most part. So.
All right. right, next question is, can you comment on the trade-off between the number of gene sequencing and sequencing depth? Would you recommend one type of test over another?
Depends on why we're doing it. Yeah, so if we're like, there's definitely a trade-off to that person's question. So it used to be more than it is now. I will say most labs kind of guarantee a minimum level of kind of thoroughness for which they look at each individual gene. So something like exome, that is sort of the historical trade-off, was that exomes are really broad look, but perhaps a little more shallow than if we were to look individually at specific genes. I think that's improved. I think there's less of a trade-off than there used to be. But really, yeah, again, it's sort of the like time, we're trying to maximize how do we get this person their answers as soon as we can, but also make sure that we're analyzing the DNA with sort of appropriate depth to say like, yes, indeed, we looked at this and it came back negative or it came back positive. So yeah, there is some of a trade-off there, and there's a little bit of like, we want to get it right. We want to get it right and do it right, and also we want to get it done as quickly as we can.
Great. Okay. And then we have another question of what are the trickiest ethical landmines in all of this? And I did ask to clarify. Yes, she says, what are the ethical landmines within gene therapy?
Ooh, there's a lot of them. And I think the biggest one that people used to worry about, and it's still kind of a big question, would be, modifying DNA that can then be passed on to subsequent generations. So our DNA that we're born with, we then kind of go on to pass on to our kids. And so there is some concern that perhaps if we're modifying that, then we're sort of introducing that into future generations without having decades of long-term experience to know that it's not going to cause any issues. So there's that one, and then also one that hasn't come up or occasionally comes up more in like popular media than really in clinical care, but what are the sorts of things that are that gene therapy, what do we treat with gene therapy? So right now we're focusing on some of the really obvious cases, like diseases where somebody may pass away early if they have it or they have significant symptoms. But as this improves over time, there may be conditions out there where some people would say this is something I would want to consider undergoing gene therapy for, but other people might say, I don't think that's something that that we might considering changing their DNA for. And I think the central question it gets to is that our DNA is what makes us who we are. It's all of the things that it's not necessarily that it defines us, but it's a very central thing it holds some clout about, it has some kind of cultural significance. They say like, oh, it's in my DNA. And so what are the things that we modify and supplement, and what are the things that we kind of consider off limits is something kind of a little bit up to us to determine what that is, and us being all of us, not just us.
All right. I am going to go back to some of the pre-submitted questions. If there are any other questions from the people that are currently watching, please feel free to either unmute yourself, go ahead and ask, or go ahead and pop it into the chat. But if you don't mind, I'll go ahead and ask a couple from the submitted questions from our registrants. The first one, can't there be more than one kind of leukodystrophy within one family?
Oh, yes. So thank you for bringing this up. And I wanted to mention, too, that Julie Cohen is a genetic counselor at Kennedy Krieger, and she gave a really wonderful webinar for ULF. I think it was last year. I'm hoping Keely that you could link it in the chat, because it was really wonderful. And she covered a lot of great ground. It was honestly kind of how it felt like a tough act to follow. So yes, there can be more than one type of leukodystrophy in a family. It's uncommon. So most genetic conditions are rare diseases for a reason. So they're rare. The odds that there would be two that would happen in the same family it's even more rare, but it's not unheard of, and it's not impossible. It's just, I would say, very unlikely. But, people don't, we always say kids don't read textbooks. They surprise us all the time. So sometimes we see, more than one thing going on. It's very unusual if it does happen, but it could.
Right. And then maybe something you can't quite elaborate too much on, but I'll go ahead and ask, how many states are doing genetic testing for leukodystrophy? I'm assuming they probably mean newborn screening. And versus genetic testing, how are the states testing for leukodystrophies today?
So I would hope that most states would have some sort of access to diagnostic genetic testing, but I agree. I think they probably were referring to newborn screening. So in my home state, we just started doing it in the last year or two. Georgia, I adopted home. I would probably have to defer that to whatever the most recent map is out there. So adrenal leukodystrophy would be the main one, I would say, that there's a lot of momentum behind newborn screening for, but there's also been a movement for newborn screening for Krabbe disease and some other ones. But I do think, if gene therapy really does take up, if it's as promising as we hoped it would be, and some of these therapies get approved, I do think there will be a push to introducing more leukodystrophies to the newborn screen, because if the idea is that catching these early is the window at which we would want to see the most benefit from therapy, then we want to catch folks as early as possible. And we've seen that with spinal muscular atrophy, which is the other disease I mentioned. Somebody in the pre-submitted questions asked about RNA sequencing. I'm happy to talk about that. I forgot to. So if you remember kind of back to an earlier slide, but anyway, so our DNA essentially has to, there's a copied form of it called RNA, and RNA is kind of the intermediary between DNA and the protein that it creates. RNA sequencing is another thing that's been a little bit in the clinical space, mostly in cancer, but it's becoming more common in the, I think it's something we're going to see used more in clinical space coming up. So RNA sequencing can be useful if the instructions in the DNA didn't yield anything, but we can see differences in that person's RNA. It tells us perhaps just our DNA analysis wasn't good enough to pick up whatever it is. So occasionally something like RNA could be thought about, but there is a lot of technical challenges with that. So it's something I think we maybe will see more of. It's just certainly some of it in the research space, but RNA is a lot harder to get a hold of than DNA. DNA we can get from pretty much anywhere. RNA, you really have to get it from the tissue you're interested in, and that's not always easy to do.
Gotcha. Okay, we have one in the chat that says, which leukodystrophies can be treated with gene therapy?
So I suppose theoretically, any of them in which we really that have been described, where the DNA that, where we, sort of understand why differences in that gene or that part of the DNA are causing that condition. So theoretically, any genetic condition would be theoretically amenable to it. It's just whether or not we would, how far we would be in developing that. Right now, I think we're focused a lot on the ones that are treatable by stem cell transplant, because we're able to sort of use the similar protocols that we would use for that and then treat the tissue that way. But there are certainly there's a lot more work that we need to do to get those up and ready for clinical trials. And I often tell families too that one of the most impactful things that you can do as just an individual person that has leukodystrophy or a family member with one is participating in natural history studies. So I know they don't sound that exciting, but we really can't get a clinical trial going, and we can't get research going about the condition unless we understand what that disease is, and also that we understand what the people who have that disease, like what about that disease would they most like to see a treatment for? And so really understanding what the disease is and what people's experience with the disease is really one of the things that we need in order to get a clinical trial off the ground. And natural history studies are really the best and most powerful tool we have for that. So if you're not part of one or there isn't one going, I think that's one of the best ways that people can, it doesn't take much, but it really can be a powerful tool.
Right. There is one, there's a couple of people that asked a similar question in the pre-submitted questions. One person is looking to get their daughter's fiance tested as a carrier for MLD. There's another person that is saying they've never been tested. How do they get started with getting tested? And then another person was asking about if they're trying to determine if it runs in their family, but they don't have access to the medical records of their family members that might have had it. So do you have any recommendations? And I will put a link in the chat to a company called Invitae that does offer leukodystrophy screening. It's sponsored, so that means that it is no cost to the families if you're in the United States and Canada, and I believe there is one more country that might be covered by it, but I will put the link in the chat. But hopefully, Rachel, you can give some suggestions on where people can get started if they're looking to determine whether they're a carrier, if they have a full-blown gene mutation, where do they get started?
So I'll start with the carrier testing for somebody's partner, and getting tested if there's a family member that also has it and you may be at risk. It's always useful to know what the specific genetic variant that was found in relatives is, and I know that's not always easy to get your hands on. So in those cases, we just kind of have to do the best we can. So sometimes we don't have it. We try to get it, we're unsuccessful. That is what it is. For folks that are interested in carrier testing, it's often something that that a prenatal genetic counselor does. So a lot of times people think about prenatal like, oh, well, I'm not pregnant, but it's a really common referral for prenatal genetic counselors. There's a tool called findageneticcounselor.com. You can just type in your zip code. And there are some telemedicine-based platforms offered as well. So basically what that genetic counselor would do is talk to you about, okay, so this person is a carrier, you're interested in having your partner tested, see if they're a carrier, just to try to understand if there's an increased risk there. So that's a really common referral to prenatal genetic counselors. There are some obstetricians and primary care physicians out there that feel comfortable doing that. I wouldn't say everyone does, so it's just kind of asking them about it or asking them about a referral for it. For folks that have a family member with it, I think it's just kind of talking to your physician or finding a genetic counselor or a genetics clinic in your area that's able to offer testing for it. And then for somebody that maybe was diagnosed with just sort of broadly with a leukodystrophy or sort of an unspecified type a long time ago, then yeah, something like that. That sponsored leukodystrophy panel may be able to reduce the cost. But even, I think insurance coverage has slightly improved for genetic testing. The technology really has improved in the last 5 to 10 years. Like it is, I meet families that are like, oh, it was so expensive and it took a year and we never even really got answers. And it was like 15 years ago. And I'm like, oh, it's a cheek swab and it takes 3 weeks. And people are like, what? So it is. So I think just kind of reaching back out to folks involved in your healthcare team and asking them about, what would genetic testing look like and how might I get a referral to somebody who is comfortable doing that. Because you do really want to do that testing in capable hands, because it's if you feel like it's confusing and you're not a healthcare provider, I'll let you know. I work with a lot of healthcare providers that are confused by it as well. So it's good to make sure that whoever you're working with feels comfortable doing it.
All right, we do have one final question that is in the chat, and then we will say goodnight and thank you to everybody for joining us. But the final question is, how does gene therapy stack up against ASO therapy or siRNA therapy?
Really good question. So sometimes, I will define what those terms are. So ASO is an antisense oligonucleotide therapy, and then the single-stranded RNA therapies, they're a little bit similar, but essentially what it does is it's using something like a DNA-like molecule or an RNA-like molecule to sort of encourage the DNA to behave differently when it's interacting with that therapy. The pros of it are that it's less permanent. It's a little bit more time sensitive. Sometimes we're able to kind of get it more directly to the tissues that need it. The cons being is that it's you do have to kind of keep it up. We don't have both of those for most diseases are still in the research phase. So I wouldn't say we have any real, like even for diseases where we have an FDA approved antisense oligotherapy and a small molecule therapy and a gene therapy approved, we don't have any real head-to-head data to know which ones work better. And sometimes it's just kind of a conversation with the treating team and the family is just saying, you know, what works well for your lifestyle and what does the data say? There are certainly pros and cons to both therapeutic strategies. They're both, again, a really exciting area of research. So I appreciate that person's question. Maybe there's another time we could talk about ASO therapies, but we don't have great data on kind of how they compare. And right now they're just a little bit different from each other. So it's about kind of balancing how they work and how do we get it to the person.
All right, wonderful. Thank you so much, Rachel, and thank you so much to everybody that joined us this evening. If you are watching the recording that we will be posting to YouTube tomorrow morning, thank you so much for joining us to rewatch. But we really appreciate the involvement of everyone in our community. We hope that you enjoyed the session, that it was educational for you and you're taking away a lot of information. If you do have any questions, please do not hesitate to reach out and ask. Feel free to email us at office@ulf.org with whatever questions you might have. I'll pass them on either to Rachel or to any of our other wonderful medical professionals that we have within our communication network. We will work towards getting your questions answered for you. But otherwise, once again, thank you so much, and everyone have a wonderful night. Thank you so much, Rachel, for being with us tonight.
Yep, thank you again. Take care.